This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1125, recorded on June 21st, 2024. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon Despommier.
Hello, Vincent, and welcome back to the show.
Thank you.
We missed you dearly. We missed you. That's a sincere comment. Rare for me, but I gave you one. Looking out the window, it looks beautiful. Looking out the window, it looks beautiful. But if you go outside, you could fry an egg on the sidewalk, so to speak. It's really hot, and it's going to get hotter. So, enough said.
I'm in New Jersey. I don't know what the temperature is, but it's pretty warm and sunny. It's a very nice day. Also joining us from Madison, New Jersey, Brianne Barker.
Hi. I'd like to echo what Dixon had to say and say welcome back.
Thank you.
It's also extremely warm here. It is 96 Fahrenheit, 36 Celsius, and I am pleased to be indoors.
Yeah.
And from Montreal, Canada, Angela Mingarelli.
Hello, everyone. So actually here today, it's a little bit cooler. I guess the warm front is moving south because today it is 24, I think, C. So it's like 75
That's a great
Fahrenheit.
temperature.
But yesterday it was like,
Nice.
for the last 4 days, it was like 105. It was very, very hot. It was like plus
Wow.
40 degrees Celsius.
Yep, that's how it's
And
gonna be here.
I don't have AC. So I stayed at my sister's house for 3 days. She has air conditioning because me and my dog were melting. So,
You could always go to a public library.
well, the lab is good. Yes, the lab is very cold. But
Right.
Mhm.
But I don't think they take dogs at public libraries, Dixon.
Or at the lab.
Right. If you enjoy these programs, we'd love to have your support. You can go to microbe.tv/contribute. Microbe TV is a nonprofit entity, and your donations are U.S. federal tax deductible. One announcement today, the 2024 International Hepatitis B Virus Meeting is taking place September 11 to 15 in Chicago. You can go to hbvmeeting.org to register for that. And I will be going and doing a TWiV with some of the participants. We have two news items.
Oh, do I not have to hold up my ASV badge this time?
Kathy said you don't have to say anything because it's too late.
It is.
A couple of
The
news
time is
items
past.
for you. It was a newsy week. Actually, it was one more I didn't put in. We'll do that next week. House Republicans fault NIH for evasive answers on risky monkeypox virus study. So what's going on here?
Yeah, what is going on?
So the Republican lawmakers, okay, I don't understand why this has to be a partisan thing, right? Science is not supposed to be that way. But the Republican lawmakers criticizing NIH. These are members of the House of Representatives Committee on Energy and Commerce. They accuse NIH of obstructing an investigation of a proposed research project on monkeypox virus at the NIAID. And basically they're saying NIAID can't be trusted to oversee its own research. These are gain-of-function studies, and they need more oversight.
So, first of all, why is the Committee on Energy and Commerce the ones that are dealing with this?
You got me.
I thought that too when I was reading it. I was like, in what world are they commenting on this or even understand what that research means?
Exactly. That's part of the problem, right?
Yeah.
It's part of the larger problem.
Anyway, the NIH spokesman said, there's no issue here. We're totally transparent. We're telling you everything. And I'm going to tell you what's going on here in a minute. So the research is going on in the laboratory of Bernie Moss, a well-known pox virologist who's been on TWiV. And as you know, there was monkeypox virus outbreak some time ago. And so the project is, there are two clades of monkeypox virus, clade 1 and clade 2. And one of them is more virulent than the other. Clade 2, which one is more virulent?
1 is virulent.
and
2
2 is
is
less
not.
virulent.
Hmm.
So they did experiments where they took genes from the less virulent monkeypox virus and put them into the more virulent to see if it would lose virulence, right? Then you
Right.
say, this gene is involved in virulence. So that's fine. They don't seem to have a problem with that. But they had also proposed to do the other way, is to take the less virulent virus and add genes from the virulent virus, which could ostensibly make it more pathogenic, right? And this committee said it could cause a pandemic.
Oh, really?
They said
Isn't that
No.
that they think they're experts on pandemics?
they say it's risky gain of function research because the virus could be more dangerous and even touch off a pandemic. And when they asked information from NIAID, they said, well, we never did these experiments. They weren't approved and we never did them. But it turns out that they were approved, but they just weren't done. And the committee wanted some paper trail showing that they weren't done. So I don't know how you do that. If you don't do an experiment, you don't write in your lab notebook, we didn't do this experiment, right?
Did not do these experiments today.
That's right.
Yeah.
I mean, you're in the hallway and you say, this might be an interesting experiment. No, I don't think it's worth doing. Okay, I'm back to
Right.
the lab. That's
Write it
it.
down.
Yeah, there are 2 other parts. Well, there are a lot of parts of this article that I was confused by. But one of the things they also mention is that when they did the transfer of clade 2 genes into clade 1, it didn't actually change the phenotype.
No, it didn't.
So why suddenly these energy and commerce representatives think they know the outcome of the other experiment is not clear. And this quote about, well, we want evidence and it could be in lab notebooks or emails or what?
That's
Are
in
they
Wuhan.
going to
It's
take
actually in
all the lab
Wuhan.
notebooks of all the people ever?
No, it's in China.
Good luck
That's,
interpreting
everyone
that.
knows
Good
that
luck.
it's in China.
What are you talking about, Dixon?
Nothing. I'm just mumbling.
You're just joking. Okay.
Just joking.
So the lab is contending that they didn't do these experiments, and the committee says you're lying, you're not being transparent because it's a political thing. They want to make a political issue of it. This is part of a broader attack on gain of function research. We'll see another document that we're going to look at today, revamping the NIH, which also deals with this. But so the people on the committee say, our gain of function guidance is not good. The NIAID should not be policing itself. So basically, if a lab at NIAID proposes an experiment, it goes through NIAID and they say whether it's a problem or not, a potential problem. And this committee says, you can't do that. You need to have an independent committee looking at what you want to do.
Who's this independent committee? Because isn't the idea you want people who are trained in the subject?
You would think so, yes. Who are actually doing the work, right? But
Right.
they think you're conflicted. You can't be neutral because you just want to do your work and get your research funds and blah, blah, blah. So no one is accountable, apparently. No one can trust anyone else. Maybe that's true in the Republican world, but, scientists, it's different. Anyway, the last quote of this paper is good. Nicholas Evans, who is a biosecurity expert at UMass Lowell, thinks the guidance we have is fine. Much of the concern about Moss's proposed experiment, he says, is based on the assumption that a modified clade 2 virus would be more transmissible than clade 1. There's no reason to think that would be the case. And that uncertainty means that the monkeypox experiment does little to inform the discussion over gain of function regulation. It's being used as a way to prop up a debate about the regulation of the life sciences, which I think is probably in bad faith. And that's just perfect quote, right?
Wow.
Bad faith. I agree. Anyway, the article is written by Kai Cooper-Schmidt. I saw the headlines even said that Bernie Moss made monkeypox in his lab by gain of function, Yeah. This is the danger of this crap. All right. Another news item. This is very interesting. Let me get to my PDF copy. Here we go. Wild polio virus makes comeback in Afghanistan and Pakistan. So as you may know, wild polio viruses type 2 and 3 have been declared eradicated. because there is no more poliomyelitis caused by those two serotypes. Unfortunately, most of the polio happening globally is caused by the vaccine type 2 strain, which is another problem altogether that we've talked about. But type 1 has continued to circulate in certain parts of Afghanistan and Pakistan. And the goal has been to try and end all transmission. And in 2021-22, they actually cleared virus from Karachi, which is amazing, is a huge city, right? And it also disappeared from two other historic reservoirs around Quetta in the southwest province of Balochistan and Peshawar in the northern province of Khyber Pakhtunkhwa, both on the border with Afghanistan. But now, wastewater sampling reveals that the virus is back in Karachi and around Quetta and Peshawar, likely brought in by people from these districts. I would argue that probably was never gone. It's just your sampling wasn't that good.
Hmm.
But the idea that you're going to stop transmission is defective because OPV does not stop transmission. It does not stop transmission. So the idea that you can stop it by this is wrong. Now, the number of polio cases is low because many people are immunized, right? But they say here, plenty of virus is circulating, ready to strike any unvaccinated kid. So they had a goal of stopping all transmission of wild type 1. This year, that's gone. They have to rethink their approach. And, this is unusual. These are unusual conditions, right? Really high population density, a lot of movement of people. About 1 million people a day move in and out of Karachi every day. That's a lot of people.
And not an easy place to vaccinate.
That's right.
No.
So 2024 target of ending all transmission will be missed. I would say you're never going to end transmission. But this is just another setback to this idea that you're going to stop transmission. Just stop the disease, folks. I think that would be enough. But no, they got stuck on this eradicate, and now they don't want to back up. But you'll see that they're not going to be able to eradicate the virus. Okay, now we have a snippet, which is there's a science article that describes it, and then there's a PDF, which is the document. House lawmakers float plan to overhaul the NIH. Let me see where that is here. Here we go. So this is an article in Science by, I don't know, the name isn't on here. Does anyone know
Jocelyn
that?
It's also
Kaiser.
Kai. Oh wait,
Kai
no.
Cooper Smith. Okay.
I think it's
Republican
Jocelyn
leaders of two
Kaiser.
committees in
Yeah,
the
it's
House
Jocelyn Kaiser.
Jocelyn Kaiser. Okay, floated a proposal to streamline the NIH and curb potentially risky pathogen research, among other reforms. Okay, so I think it's fine to streamline a big agency like the NIH, but what is this potentially risky pathogen research? I've never known a pathogen to escape a lab and cause an epidemic or a pandemic. Have you?
No, Vincent, this may be all a reaction to the fact that virtually every year that the budget for NIH comes up, the Republican portion of Congress turns it down, and then the president overrides it and gives even more money to those agencies. So this may be a reaction to that.
Yeah, I will note that almost all of the politicians who are cited here are Republican politicians.
Yep.
In fact, they are members of the House Energy and Commerce Committee, who we heard about before,
There you go.
Scientists.
who don't want to give money for commerce, I guess, to the research.
So this is of interest to us because most of us have received NIH funding for our research over the years. It's the premier biomedical funding agency in the US. And, you have to make sure that what you do is not going to break it, right? So the document, which you can get, Reforming the NIH Framework for Discussion. The time is now to build a stronger NIH for the future. Okay. What I find interesting, so there's a whole bunch of history on the NIH, and they have a list of the current NIH institutes. So maybe not all of you know, there are many institutes at NIH that do different things like NIAID, National Institute for Allergy and Infectious Diseases. There are 27 different institutes, and collectively they get a total of $48 billion in federal funding. Things like National Cancer Institute, National Heart, Lung, and Blood Institute, National Eye Institute, National Institute of Aging, National Institute for Allergy and Infectious Diseases. Okay, so they want to reorganize that to 15. They want to go from 27 to 15. So, for
And it was
example, there
interesting.
will
There's
now
only
be. Sorry, go ahead.
Go ahead, Vincent. No, I thought it was interesting. I was looking through. There's only one, which is NIAID. What is it? NIAID?
AID is allergy infectious disease.
Exactly. And it's the only one that's actually going to be split into two institutes, into the separately infectious disease and then immune system and arthritis.
Right.
Yeah.
That was interesting.
Which seems strange because they're like, oh, it has too much power, but it's not the biggest one.
Yeah.
No, it's not.
And only one of them is losing funding and losing a very, very significant amount of funding. Almost $1 billion, $973 million, the National Center for Advancing Translational Sciences, Biomedical Imaging and Bioengineering, and Common Fund. I don't know what that means. Did you guys see that? It's just Common Fund and whatever that means, but they're losing almost $1 billion. And they're the only center that's losing money in this, at least from what I can see. Does anyone know why?
I don't know what that is at all, no.
Can hypothesize? Okay.
Because it's not on the new reorg. It's gone, right?
Yeah.
No more Common Fund. There's also a director's budget. You know, the director of NIH has a budget of $1.9 billion. I don't know what that's for. But, you know, what do you think about making a National Institute on Infectious Diseases and a National Institute on the Immune System and Arthritis?
They usually say don't try to fix what isn't broken. And I don't see either one of these concepts being separated. I mean, even the department that you and I were in, Department of Microbiology and Immunology, you put them together because so much of immunology is directed against infectious diseases.
Sure, of course.
Well, exactly. And arthritis, couldn't that be part of bodily systems? The one that says body systems research is kind of weird to like throw out arthritis.
Yeah, why they picked arthritis compared to a variety of other things that come up with the immune system, I was confused by.
Well, you can catch arthritis from a tick.
Yeah, it's like it's an inflammatory. It's inflammatory, but I mean, like, so are many other things that are in the bodily systems. When you could have they also have arthritis in there, though. So it says National Institute of Arthritis and Musculoskeletal and Skin Diseases is becoming part of this Body Systems Research Institute, but then they have arthritis separate again with the immune system. I don't understand because they have it included in the other institute already. It's kind of redundant.
There are many causes of arthritis.
Yeah, true. The National Institute on Body Systems Research is sort of an interesting one in that they're combining heart, lung, blood, arthritis, musculoskeletal, skin, diabetes, digestive, and kidney. I know of immunologists who have submitted to all of those, and so why immunology is different than body systems is a little weird.
Yeah, it's kind of odd. And I don't know, because technically immunology can be implicated in any of these, literally. But
I think a congressional committee looking at the NIH is like the blind man and the elephant, because none of them actually knows what's going on underneath those doors. They've never been in a laboratory. They've never participated in any research programs. They're just reorganizing for the sake of saving money so they don't have to pay an institute to do the work that they're doing now.
I think the bottom line is the same.
The bottom line
$48 billion is the budget. 48,174. It's the exact same. It's just been moved around.
Well, the budget is next. That's
Anyway,
next week's article.
So they give a little history of NIH. Did you know that the NIH, the mission is to seek fundamental knowledge about the nature and behavior of living systems and the application of that knowledge to enhance health, lengthen life, and reduce illness and disability. So there's a little history here, which I won't go into, but did you know it started in 1887 on Staten Island? I didn't know that.
I did not know that.
Yeah.
Cool.
Now there are a bunch of bullet points. the recommendations that I wanted to go through a bit here. Let's get to them. They have a graph of NIH funding, how it's gone up over the years. It's very nice. We should be investing at NIH. It's a good return. Okay. So, recommendations. Restore Congress's role in directing funding. So, right now, Congress doesn't have a say in what gets funded. They want to have that back.
That is exactly right.
They want to read my grant?
Yeah, I was going to say how they won't even be able to interpret what they're reading necessarily.
No,
Like they
will
won't
say
understand that.
Brienne is in a district with a Democratic representative. We're not going to give Brienne her money. Crap like that. I mean, how would they know? How would they know what to do? So that I have a lot of problem with this restore
ridiculous.
Congress's role in directing funding. I mean, historically, when I came into the R01 business, there was no congressional involvement whatsoever. And then I don't know if you remember, who was the guy Dixon who talked about the Golden Fleece Awards? Remember him?
Oh, yeah, he was from California, I think.
He would pick out grants and say, "Why are we funding this? Why are we funding research on a worm or a fly? What does this have to do with
He
human?"
had a hair transplant at one point. I know a lot about him except his name.
So he called them the Golden Fleece Award because he felt
right,
these scientists were fleecing the government.
Exactly.
And I'll bet if you looked at some of the ones he complained about, they have all led to very important findings. Right?
Exactly.
I know, like, why would you study the immune system in prokaryotic cells?
Yeah, exactly.
You couldn't. I couldn't think of a reason why that would be useful.
No, not at all.
All right. So another bullet point under this part is to re-examine indirect costs. So for those of you who don't know, when you get a grant from NIH, you get $100,000 for your research, and then your institution gets what are called indirect costs. You don't get them. They go to your institution to pay for the lighting and the toilet paper and cleaning the floors and so forth, right? Overhead. And the indirect cost rate varies according to the institution. Everybody negotiates their own rate.
Yeah, and not only that, it's determined by the Office of Naval Research, which I never understood that one at all.
That's right. That's right. So they want to figure out if there's another way we could do to tie the indirect rate cost to a percentage of the grant award instead of making it per institution. Part of the issue is there are universities like, say, Columbia that have an endowment, right? So they can pay for things. But then there are research institutes that don't have an endowment and they need actually more. So in some cases, their indirects can be over 100%.
Right.
So every dollar you get, the university's getting maybe $1.50. So, Congress doesn't like that and they want to fix that. But the universities, the institutes are going to be very stubborn about changing that because that's what makes them support their the infrastructure, right?
Yep.
Understandably so.
Okay. Now the next one. Grant reform. NIH grant research must protect against national security risks and threats and be independent, innovative, responsive, and transparent. And this is where gain of function research comes in. And they write here, there's been increased concern about NIH approval, management, and oversight of gain of function research and dual use research, particularly that involves pathogens with enhanced pandemic potential. Gain of function research is used as a broad term to encompass scientific inquiries where an organism gains a new property or an existing property is altered. That's a good definition, right? I can take that. This includes both naturally occurring and experimentally induced changes in organism to better understand transmission, infection, and pathogenesis. A subset, sometimes called gain of function research of concern, GoFROC, okay,
Such a
often
weird acronym.
involves experiments that enhance a pathogen's transmissibility or virulence or disrupt pre-existing immunity. So, for example, if you make a virus resistant to a monoclonal antibody and that virus is a human pathogen, that could be construed as gain of function of concern, right? And This all started with the H5N1's transmission studies back in 2015, which we covered extensively on TWiV. That two laboratories made H5N1 transmissible among mammals, which it hadn't been, ferrets in specific. And people were like, whoa, why would you do that? And neglected to point out that when it gained transmissibility in ferrets, it's lost all its virulence, right? That was just not even mentioned in the dialogue. So that started this whole thing that we need more oversight of gain of function research. Now, they do point out there is a scientific community that says we need to understand viruses, right? We need to do these kinds of experiments. So you can't not do them. And then there are other people who say we should never do them and just forget it, which, there has to be some in between, right? Because you can't just not do experiments on H5N1. And they bring up the monkeypox virus experiments that we've talked about here. They bring up the Eco Health Alliance NIH grant that was used to support coronavirus research in Wuhan. They say it was mismanaged, and technically it was. Apparently, they didn't file some reports that they were supposed to file, but experimentally there was no mismanagement. The experiments were done properly, so it's all a technicality essential. So basically, their idea is that NIH, in particular NIAID, doesn't have a good process in place to weigh the risks and benefits of such experiments. And so they propose that we have a committee that does this. And they have some recommendations, which I find really interesting here. First of all, grant recipients must remain dynamic. Provide grants and award only to primary investigators that do not have more than 3 ongoing concurrent NIH engagements. That's going to piss a lot of people off because there are a lot of people that have more than 3 NIH grants, big labs. And so I don't know what the point is when we're having a discussion here about gain of function research and safety. What does having more than one grant have to do with that? I don't get it.
I think they don't want people to have a particular amount of power. If you have many NIH grants, then you're directing a lot of what's going on in research.
Yeah. Another one, continue prohibition of risky gain of function research. Prohibit NIH from conducting or supporting certain risky gain of function occurring in countries that have been designated as foreign adversaries, and pause any such gain of function research of concern until a thorough, comprehensive policy with appropriate guardrails to monitor research that has the potential to pose risks to public health and national security is enacted. All of this is started from the avian H5 transmission experiments and, of course, more recently, SARS-CoV-2 origins, right? The vast majority of Congress thinks this virus came from a lab, and they're just wrong, as we pointed out on our episode a couple of weeks ago. But it's made into a political issue because it's convenient to do so, right? But now they say, oh, this is dangerous, risky research. We have to regulate this gain of function. You can see that this virus may have come from a lab. This is not good. It's just, it's wrong. It's all wrong.
It's wild when you think about it, where they pull all of the grants and all of the funding for viral discovery. So to actually try to see what's out there in the wild, sampling animals, sampling wildlife, and then also the viruses that we do know exist, we can't even learn from them now. So either way, it sounds like, I don't know. It just seems wild that we don't know what is out there. And then what we know is out there, we can't even understand at this point because people can't get funding for for doing experiments on these viruses. Yes, there's obviously levels of different ways you can manipulate viruses, but I mean, just saying that everything is gain of function and stopping all gain of function research within funding from the NIH seems crazy.
I mean,
well, there may be some undertones of religiosity associated with this too, because if you look back in history to the great scientists of the past, particularly in the 16th and 17th century, even Isaac Newton was criticized for doing experiments rather than allowing God to have its way with whatever. And the mathematicians were also criticized for their role in revealing something that God had kept secret for so long.
I think in this case, it's more ignorance than anything else. That
yeah, well,
people believe
ignorance is bliss, right?
Not true, but people believe 2 or 3 scientists that say that SARS-CoV-2 came from a lab, and now everybody thinks that we're all creating these pandemic viruses or potentially creating pandemic
Right.
viruses, and it's just wrong.
Correct. I agree with you.
You know, that New York Times opinion piece did a lot of damage,
Yeah.
right? It has a broad readership. And even though we got a lot of nice emails from people saying we did a great job, they said, unfortunately, the Times is far more read than TWiV.
That's true.
So this is damaging. I don't understand why people have to do damaging things.
Or why the New York Times would even, I don't know, why they would even publish that.
Exactly. To be controversial, to get
ridiculous.
eyeballs, basically. That's what they do.
They
Right.
they sell newspapers. That's basically what they do.
Okay, then point number 2, continue prohibition of risky gain-of-function research. Prohibit the NIH from conducting or supporting certain risky gain-of-function research occurring in countries that have been designated. I already read that. All right, fine. And then the third point is the one I wanted to mention. Establish independent review entity for the proposed National Institute on Infectious Diseases. So remove final review and approval authority for certain risky gain-of-function research from the proposed National Institute on Infectious Diseases and empower a public independent oversight entity to review, modify, approve, or reject, as appropriate and oversee such research and experiments. Who's better to review the science than the people doing it?
Absolutely.
Yeah, who do they mean by the public? What does that even entail? What does that mean?
Public independent oversight.
Like, what does that, who?
Can I be on the public oversight?
Now, wait a minute
scientists?
Before
you
add too quickly, at Columbia University, they had a public advocate present in almost all the committees, including the IRB and lots of others. And I knew that person, and he was a very nice man, didn't know anything about science. All he was doing was listening for ethical issues to be transgressed so that he could address them.
Yeah, no, that's great. And I think having, an outside person is very useful on many committees. But my read of this is not that it's have an outside person involved. It's have some outside board.
Right.
You don't get to vote. You can listen, but you don't get to vote.
I just wonder the board should be scientists, right?
Absolutely.
And so who are you going to get to do that? I don't get it.
Wait, there's a society out there. Maybe they can draw on the Scientology Society.
Right. Right. Anyway, there are a bunch of other recommendations which I think are innocuous. Those are the ones that really bother me. The others I think are fine.
They're probably trying to bury the issues within a context of something that looks plausible. And if you pass that whole thing, you'll regret it.
I mean, the thing is, this is not really a bipartisan committee.
No.
Right. And so it just looks like it's a political issue. Why do we have to do that?
Vincent, what is the outcome of this, though? What are they going to do? Ask for a vote in Congress?
Let's see what the outcome.
They're already outnumbered.
No, it sounds like it's a proposal and they're talking about whether it could be part of the next Congress next year.
Right.
Next year.
2025.
Yes. When the numbers flip to the Republicans, perhaps.
But this article by Leslie
Jocelyn Kaiser.
It's okay. The lawmakers also want to pause so-called gain-of-function research at NIAID, studies that manipulate viruses in ways that make them a pandemic threat until stronger oversight policies are in place. Some Republicans worry that gain-of-function research in Wuhan created SARS-CoV-2. Why don't they listen to TWiV?
If they're so smart, then they follow the science, and if they understood the science, they know that it didn't come from a lab.
But more importantly, why is it that the Republicans feel that way and the Democrats do not? That makes zero sense, right?
Well, it does if you look at it as a political football and nothing else.
Anyway, so much of it I think is fine, but there are these few points that I think are really ill-advised. I don't have a problem with streamlining, but starting to meddle in funding decisions and reviewing proposals. We're not talking about a lot of proposals here. I think somewhere I read maybe 100 proposals a year would need this extra review. So this panel, whoever it may be, I think I'm going to nominate you for it, Dixon.
Thanks. I'd be glad to serve on it if I'm still alive.
Why wouldn't you be? It's only a few months away.
One never know, do one?
One never knows. That's right. All right.
I think reorganization sounds fine, but maybe ask some people at the NIH and some scientists what makes sense.
That would be advisable, I would think. Yeah.
At that point, they might get good advice, and then what would they do with that?
Well, you have transformed a scientific process into a political one now. That's what this is, in my view.
Right.
People can say I'm wrong, fine.
No, you're right.
But the scientists need to make decisions about funding. And if you really think that's a conflict of interest, then you don't understand how science works.
Right.
The scientists understand it,
That's
and not a lawyer from somewhere on this committee. They're not going to understand anything. They're not going to understand the nuance. Okay. And finally, we have a paper.
Right.
Let's talk about viruses.
Talk about viruses.
Nature Communications. Potential pandemic risk of circulating swine H1N2 influenza viruses. So this is from, let's see here. First author is Valerie Lesage, and the last is Seema Lakdawala, who's been on, I don't think she's been on TWiV, but we did her first paper years ago. And they are at University of Pittsburgh, Scripps Research Institute, Emory University, National Animal Disease Center in Ames, Iowa. And this is all about swine influenza viruses. As you know, wild birds, aquatic birds are reservoirs of influenza viruses, but so are swine. Swine that we raise for food consumption. And swine can be co-infected with multiple influenza viruses, and they can reassort and make new ones. Really better than anyone can do it in the lab, folks.
They are the lab.
So
I would be up for no more pig farming if they wanted to just, stop all pig farming.
That's right. We have
hey, with me. I agree.
Anti-porcine in the group.
That would solve it, but there are too many countries dependent on swine for protein, right?
It's true.
But if you remember the 2009 H1N1 pandemic virus, right? That came out of pigs after a bunch of other viruses, including human influenza viruses, avian influenza viruses, pig influenza viruses. They all reassorted, and out came a novel virus that caused a pandemic. So this paper deals with how do we assess the risk of these viruses that are circulating in pigs? And there are lots of viruses, influenza viruses circulating in pigs. In swine, we have 3 endemic subtypes of influenza virus. SWH1N1, so the SW means swine, SWH1N2, and SWH3N2. And they have been equally detected over the last 4.5 seasons. In the U.S., the H1 classical lineage is 1A, is divided into clades, a bunch of different clades, which you're going to hear about in this paper. Since the 2010-2011 influenza season, there have been 18 H1N1, 35 H1N2, and 439 H3N2 infections in humans with variants of swine origin in the U.S., including 6 from the alpha H1 clade and 21 from the gamma H1 clade. So those are not huge numbers, right? But pig to human transmissions occur frequently.
They're probably in people working with pigs.
Yes, as well as children who kiss pigs at fairs, right? Agricultural fairs. Right? You shouldn't kiss the pig, but people do.
Actually speaking, this is like a little bit of a rant, but I mean, it's not
It's
about
okay.
pigs. It's about a deer, but it's also about a child kissing an animal that then got very sick. So like you said that, and it reminded me, it jogged my memory that people that get close to deer and animals and things like that in the wild, please like be careful, because I was at a conference recently and they were talking about tuberculosis, and this woman came and she gave a talk. Well, she has tuberculosis and is currently being treated with tuberculosis, and she actually got tuberculosis from a deer. So she didn't, in the beginning, they didn't know where it was from. It was here in Canada. They were trying to figure out like how she got tuberculosis. And then she had remembered that she had been interacting with a deer multiple years before, like 2 or 3 summers before. She was feeding a deer every day. And then her grandmother had told her that, the deer looked ill and thin. And maybe that's why she was like coming to feed. Like it was docile enough to come in close to humans. So like, please, everyone, don't get that close to wildlife, especially ruminants. Yes, they can have tuberculosis. You can get sick. It can transmit to you. And unfortunately, it is a very, very, very terrible disease to have. So with pigs, and if you're working in a pig farm, then hopefully you're wearing masks and, testing and everything like that. But even with wildlife, like all the time in a forest, people are like, oh, animals are so cute, and oh, let's go like pet this animal. Okay, well, if it sneezes on you and it has like mycobacteria, well, you could get sick. So please don't.
Well, the other danger, of course, is that you might get gored by a bison.
But that's awesome.
I
wouldn't get near a bison, but yeah, deer is like more cute, and people, children want to pet deer and things like that.
Yeah, and
but back to pigs.
If
There is a
any of
petting
those
zoo.
animals are allowing you to be close enough to them,
exactly,
there might be something wrong.
They're
There are
probably
petting
ill.
zoos, right? So you want to eliminate all the petting zoos also?
Well, I mean, those animals are normally bred in captivity. They could still carry like some of these pathogens, but they're bred in captivity normally, and hopefully tested. But we don't know that either. So steer clear of animals, wild animals, especially if they look sick.
I think we should steer clear of humans as well.
Also,
right.
Politicians,
they all look sick.
steer clear.
Politicians.
Steer clear
All
of
right, so
sickly deer and politicians.
these influenza viruses in pig, they are reassorting with human influenza viruses and avian, plus they're undergoing antigenic drift as they circulate in pigs. So this all allows for the generation of viruses that could have pandemic potential. So to try and get at that, so how can we assess the pandemic potential? The WHO has made a risk assessment tree. They have basically a number of questions that you ask about each of these viruses. Can it replicate in human cells? Can it be neutralized by antisera in humans? Can it cause disease in ferrets? Can it transmit and so forth? And then you decide if it has pandemic risk or not. That's what they do in this paper with 2 swine influenza viruses.
I really like this paper. I thought it was.
It's
I don't
quite
know.
nice.
I thought it was well laid out.
So if you're wondering what people are doing, we're not just sitting around waiting. They're looking. They're saying, "Oh, these viruses are risky," and you'll see some of them are not, and some of them are.
Yeah, I liked this decision tree in particular.
Yeah,
I was like, oh, this is a nice way of analyzing this and laying it out.
so they're
So
going to look
Vincent,
at.
just to be clear, has any pig outbreaks been linked back to exposure from a human that had influenza?
Have there been outbreaks in pigs from human viruses?
Yes.
I don't believe so.
Okay.
I could be wrong, but
that would also be very hard to identify,
Um,
right? How would you? I mean, if you mean like a worker at a farm, for example, it would spill over into the pigs, and then you would
that's what I was
identify it.
talking
But then they would need to know that they were even infected with influenza in the first place, that human, and then also know which influenza. And I don't know how easy that would be.
Do the pigs have symptoms?
They can have symptoms with influenza.
Yeah, sure.
They get prostrated. They get rhinitis. Like kind of similar. They also have like
So there are
mucus,
certainly
etc.
lineages in pigs, Dixon, that originated from people.
Okay.
But I don't know about outbreaks. Okay.
Okay.
It's a good question. In fact, one of the authors on this paper, Amy Vincent, gotta love that name, right?
Amy Vincent. I was going to say.
I was going to have her on TWiV in Baltimore a couple of years ago, where there was a flu meeting, but her flight got delayed. But I'd like to have her on because she's interested in the swine human interface. We should have her on. All right. So here they're going to do a pandemic assessment, risk assessment with two of these swine viruses, the gamma H1 and or gamma swine H1N1, and then an alpha H1, alpha swine H1N2. They're going to go through a battery of tests to say, is this a pandemic risk that we should worry about? And we'll talk about what that means at the end. So the first thing they do is ask if humans have antisera that neutralize infection with these swine viruses. So they collect sera from healthy adults in Pennsylvania in the fall of 2020, and they do hemagglutination inhibition and neutralization assays. So these are people who, presumably got vaccinated against various influenza viruses, maybe also naturally infected. They're just saying if any of those antibodies going to be active. And they found antibodies that neutralized the H1N1 pandemic 9 strain, which is the one that caused the pandemic in 2009. That's not surprising because we vaccinate against that. And also gamma swine H1N1. Right. So this is a virus that it does infect people as we have said, but it's pretty limited. Yet people have sera that neutralizes. So it's probably a cross-reactive antibody response. But alpha swine H1N2, no neutralizing antibody in any of these cohort. So the viruses are different enough so that they don't react with neutralizing antibodies.
I like how they did different birth years here.
Yeah.
Because people may have been first infected with different types of influenza based on when they were born. I know there was some discussion about pandemic potential of different influenza viruses based on what your first flu experience was.
Right.
And so here they showed that first flu experience doesn't matter in this situation.
Right. So basically what we find is that this gamma SWH1N1 is of some pandemic risk. It moves further along in the decision tree about pandemic risk. But they said the other one, alpha swine H1, we decided to push it along at the same time as well, just to see what would happen. So both of them are going to go to the next step, which is to look at the gene sequence of the HA gene and look at them. So they have the 2009 pandemic virus and then these two swine viruses. And one of them is more distant than the other. And the one that is more distant is not neutralized by antibodies that are present in this cohort of people. So that's probably why. There are probably changes in the HA protein that are blocking antibodies from reacting with them.
Vincent, should we explain? Because I know we want to explain as much as we can. And I know recently on another paper, a few months ago, we did influenza, but what this H and this N and HA and all of this means for the listeners out there.
Yeah,
So H.
Would you like to do it? Go ahead.
I can do a very brief description. So H, hemagglutinin. So hemagglutinin and neuraminidase are two surface proteins on these viruses, basically on the influenza virus, and they elicit an immune response. So they are, I guess that's the easiest way to put it. They are they are immunogenic, and then there can be different types of these on different viruses. So when we say like H1N2 or H5N1, it's a different neuraminidase, different hemagglutinin with different virulence. So they're virulence factors, I guess. Is that good enough, I guess? Would you add anything? That's just like very briefly what they are for the listeners.
They also looked at the receptor specificity of these hemagglutinins. We know that avian viruses, human viruses tend to prefer alpha-2,6 linked sialic acid. So it's a sugar that the HA binds to in a certain chemical configuration. And the alpha SWH1N2 receptor specificity was clearly alpha 2.6, so well adapted to human receptors. That's important because if it's alpha 2.3, it's less of a pandemic threat, right? Because it's not going to be binding human receptors. They use the word fitness a lot in this paper, which I'm very happy to hear. Ever
I
since
saw
we
that
started
and thought of you.
talking about fitness during the pandemic, some people have listened, right? And just think of changes in a viral genome as affecting fitness without you knowing exactly what that means. So they said, let's assess fitness of these swine viruses for cells of the human respiratory tract. So they make cultures, human bronchial epithelial cultures at an air liquid interface. So these cells differentiate into the proper air liquid epithelium that you have in your respiratory tract. And they infect with these different viruses. So they include the 2009 pandemic and then the gamma and the alpha virus. So the gamma replicated half as well as the 2009, and the alpha replicated about 70 to 80% as well. So they say that's good, that's comparable enough. Despite all the amino acid changes in the hemagglutinins, um, these viruses reproduce well in human cells, and that moves it along the pandemic risk decision tree.
Yeah. So at this point, we found that these viruses, the alpha virus, is something that we don't already have immunity to. Is something that seems to be able to interact with human cells based on multiple measures.
Right.
And so, just in vivo types of things, this virus looks bad, or potentially
Concerning.
not good.
Potentially concerning.
Yeah.
All right. So, again, no matter no matter what the amino acid differences, they replicate pretty well in human bronchial epithelial cells, which is where the viruses are going to be reproducing in humans. So that's a concern. The other part of the decision tree is transmission, right? So if you're infected with an influenza virus, to transmit it to another person, it has to be expelled in respiratory droplets, right? So they they make droplets of viruses from these cells in culture, and they expose them to different relative humidities because we know that relative humidity controls the ability of influenza viruses to replicate. At high relative humidities, the droplets that you expel take on water and they fall to the ground quickly, so they don't go to the next person. But under low to moderate relative humidity, they remain suspended longer. They found that the 2009 virus and the alpha swine virus propagated in these bronchial cells had very little loss of infectivity at all the relative humidities tested. So basically, they can be transmitted over a wide range of relative humidities, which is another concern on this decision tree. Another part of the decision tree is the acid stability of the HA. So after the HA binds the cell surface receptor, it's taken into the cell by endocytosis. And in the endosome, the pH drops, which leads to the viral membrane fusing with that of the endosome, and that's how the viral RNA gets out. And so they wanted to look at the pH stability of the HA as kind of a surrogate for fusion because they're not doing actual fusion assays. So they just basically ask if the viruses are stable over a range of pHs. And in fact, the alpha swine H1 has a pH of fusion comparable to human influenza viruses, which is below pH 5.5. So it looks like fusion would not be an obstacle for these viruses in human cells. The other important part of this is the neuraminidase, the other spike protein on the surface of the virus. You need to have a balance between HA and NA. The NA allows the viruses to move away from the cells and be transmitted. So they measured neuraminidase activity of these swine viruses, and they found the alpha swine H1 has similar neuraminidase activity as the 2009 pandemic virus, which they say is a feature consistent with a virus capable of airborne transmission. And they always say, and requires further characterization. They say that all the time. We need more money to study this. Okay, now we go into ferret models to study transmission. So in the ferret, human seasonal influenza virus transmit between ferrets 2 days after exposure of your first ferret. So if you infect the ferret, 2 days later, the human influenza viruses will transmit within 2 days. So they infect ferrets, which we call donors, and then they put them in a cage with recipients with a divider in between so that any transmission will be by respiratory droplets, not by ferrets, touching each other, which they like to do. And how you know that transmission occurs is you have virus in the nasal secretions of the recipient ferret, or seroconversion. The ferret makes antibodies to the virus. All right, when you do this, 4 of 4 recipients without prior immunity shed alpha swine H1N1 2 days post-exposure. So that's consistent with human transmission. So alpha swine H1 efficiently transmits to animals without prior immunity within 2 days. Remember, these animals are seronegative. But then they ask, what's the effect of immunology on these transmission? Immunology, which has been
Go team immunology.
which is going to be taken out of the NIAID.
Yeah. And put with arthritis, which is also immune mediated sometimes. But
So you have 4 recipient ferrets. They're infected with H3N2 or H1N1. Then 4 months later, once the antibody response, well, they say the immune response has waned, then they expose them to infected donors for 2 days, in particular the alpha swine H1. So they wait because if you do the experiment 2 weeks after immunizing them, you're going to have very high antibody and T cell levels, and it's not going to be a good reflection of what's going on, right?
Right.
So they take these infected ferrets and they expose them to alpha swine H1 donors for 2 days. In one experiment, 4 of 4 H3N2 recipients shed alpha swine H1 at 4 days, and 2 of 4 shed in their second replicate. So that's H3N2 would be a heterologous virus that shouldn't give any cross reactivity. Only 1 of 4 recipients of the H1N1 pandemic 2009 virus shed detectable levels of alpha swine H1N2, and all these animals were infected, which suggests that the alpha swine H1 can transmit to animals with prior immunity, which puts them in a higher pandemic risk category. Although we don't know yet if they could spread them, we just know that they're shedding them. We're going to do that in the next experiment. Okay, it's a little bit complicated. Hopefully you're following. Okay, next, transmission, of course, is a part of the pandemic risk assessment. So they asked whether ferrets who received the virus could then transmit it onward to naive recipients. So they have 2 different change where they have 4 alpha swine H1 infected donors. They're exposed to H1N1 2009 recipients in the adjacent cage for 2 days. And then those are transferred to another cage to see if they can infect another recipient, right? So you want to establish a chain of transmission. So in the first replicate, 2 of the 4 ferrets shed alpha swine H1N2, whereas in the second, all 4 had alpha swine H1N2 in their nasal secretions. Most of this shedding was on day 3, but only 50% of the infected donors transmitted onward to another ferret. So even though they're shedding, it's probably not enough to transmit to another ferret.
So that's good, at least, that
Yes,
some pre-existing immunity may block transmission potential.
that's right. That's the conclusion. Onward transmission of swine alpha swine H1 is possible even in the context of pre-existing immunity. So it's got a higher risk potential. Then they looked at the pathogenesis of infection with this virus. So no difference in alpha swine titer was observed between H3N2 infected ferrets and those with no prior immunity. However, the H3N2 immune ferrets cleared the virus by 5 days. So there's apparently some cross-reactivity. Ferrets with pre-existing H1N1 2009 immunity shed less virus on days 1 and 2 and 3 compared to ferrets with no immunity. They found robust replication of the virus in the lungs in ferrets that were not immune, the lungs, trachea, soft palate, nasal turbidus, whereas the H3N2 immunized ferrets had only detectable virus in the soft palate. That's alpha swine H1N2. And these ferrets immunized with H1N1 2009 had completely cleared the virus from their respiratory tract on day 5. Next, H1N1 immune and non-immune ferrets were infected with alpha swine H1N2 and sacrificed on day 3. They could find detectable virus in the respiratory tract, although the titers were less than in animals without prior immunity. So to summarize all this, prior H1N1 2009 immunity can reduce the viral load in the ferret respiratory tract and decrease the time to transmission to clearance of alpha swine H1N2. And remember, 2009 H1N1, the pandemic virus, came from pigs. So this is not surprising that there's some cross-reactivity, right? And then they do some pathology to compare pathology of these viruses. They basically find that pre-existing immunity can reduce the pathology. Pre-existing immunity to 2009 H1N1 can reduce the pathology caused by alpha swine H2N2 infectin. And finally, clinical outcomes. So intranasally alpha swine H1 infected ferrets with no prior immunity or with H3N2 immunity had similar symptoms, weight loss and other signs and so forth. While the intranasally infected ferrets that had been immunized with 2009 H1N1 had no symptoms whatsoever. So the summary is that although alpha swine H1N2 can transmit between immune animals, its replication and severity is limited by prior immunity. So that's the decision tree that you do here. A couple of points I want to make. First of all, they mentioned that protection against influenza virus in hosts without antibodies can be conferred by CD8 positive T cells, which recognize conserved influenza virus proteins, right? So many of these viruses likely have conserved proteins other than the HA and the NA. It may not prevent infection, as they say, but it will more efficiently allow clearance of virus and faster recovery from illness. And in fact, that's what they saw in their experiments. If they immunized with H1N1 2009 and challenged with alpha swine H1, you get faster decrease of shedding and fewer diseases. So it's very cool. So swine H1, alpha swine H1 has a higher pandemic risk than gamma. And they say we should keep doing surveillance to capture zoonotic events, which means let's see if this spills over into people, right? And maybe we should consider vaccinating pigs against this particular virus, right? Since it
That would
has
be
some of
very
these.
expensive.
Yeah. Probably you won't convince the farmers to do that, right?
Yeah, that
Because
might be
it's to
hard.
protect people. It has nothing to do with the pigs, right?
Mm-hmm.
Yeah, it's unfortunate.
And it will probably raise the price of pork also.
Yep. But it would be the right thing to do to try and decrease the virus circulating in pigs, right?
You bet. You bet.
Anyway, if you wanted to know how you do risk assessments, those are the experiments for this particular swine influenza virus. So it was pretty interesting. Let's do a couple of email. Dixon, can you take that first one?
Sure. John writes, Vincent et al. I had heard about the honeybee disease foul brood that kills off the entire hive, but didn't realize it was a bacteria, Paenibacillus species infection. But the rule, that is, if there's a bacterium, there's a phage, for it applies, and while phage therapy aimed at human diseases is nothing new, there seems to be complications with its deployment. But honeybee hives appear to offer a much simpler system than humans for introduction of phage into, and my old department's Heather Hendrickson, now in New Zealand, and mentioned in an earlier letter as co-organizers of the VOM meeting, is behind such an effort there. And phage to the Paenibacillus to the bee's sugar water. In other words, you add the phage to the bacillus, and then you add the bacillus to the sugar water that the bees feed on, and they will wind up incorporating it into the hive. Apparently, this is not a novel idea, but in New Zealand they had to develop their own phages because of import restrictions and because of a prior collection that had been destroyed. So the second link contains details on phage hunting there, and that involved beekeepers from all over New Zealand and validation of the positive isolates. Sounds like a lot of work. Here's the version for popular consumption. Apparently, it was a news item published in New Zealand. And here's the publication behind it, and he lists that. Meanwhile, a lovely afternoon yesterday when I started this in the lower 20 Cs in Greater Braddock after drying out from the rain that came Friday during and after a warning of tornadoes that largely didn't materialize. Where is John from anyway? He's not from New Zealand, is he?
No.
No.
Um,
Braddock, England.
Greater Braddock.
Where is Braddock?
Let's see.
Hey, this
Isn't
has
that
come up.
Texas?
This has come up before. Pennsylvania.
Pennsylvania. Okay.
So just to clarify, the Paenibacillus is causing infection in the
Yeah,
honeybees.
right.
So they add the phage to the drinking water of the honeybees, and that takes care of the infection.
Right.
That's very cool.
Yeah.
Phage therapy for bees.
I love it.
So you skipped the first one, but I'll read it.
I saw that. Yeah, Dixon, there was a one-liner, two-liner.
Shirley writes, you have credit lines for everyone participating in TWiV, including timestamps and music. Who's responsible for the puns, the titles? Credit and blame must be given where blame and credit are due. All right, so I don't understand the rest.
I do.
Part of it is great.
Alan AD says,
Alan
surely you must be joking. That's from a movie called Airplane, I believe.
Really?
And
the person's name who wrote it is Shirley.
And he was telling jokes, and so he said, surely you must be joking. And I wrote the second one, ARG times 2, because I couldn't get to the second power over ARG. I don't have those instructions up here above me on my editorial list.
So the titles are mostly Alan, but not always.
Yeah.
Sometimes others make titles too, like other people, right?
Yes.
Yeah, we discuss at the end of every episode.
Yeah, after we stop recording,
he really has a knack for it.
We discuss titles and we come up with things. I always want the straightforward titles so that people know, because a lot of people write and they say, I don't know what the episode is about if you make these clever titles. So there's an argument for that, right?
There is.
But we still have some clever titles. All right. Brianne, can you take the next one?
Sure. Patrick writes, Dear Twivers, I cannot overstate the influence you and your podcast have had on me. Like many of your followers, I joined the viral bandwagon during the early days of the COVID pandemic. I was working as a chief resident of the UC San Diego Pediatrics Residency Program, where I completed half of my MedPeds residency, and Victor Nizier recommended TWiV on a well-attended university-wide town hall. I have been a devoted follower since, at least when my work schedule allows. And when I moved to the southern tier of New York State to work as a hospitalist, Daniel's weekly clinical updates helped me stay up to date on COVID as our hospital was inundated with sick patients in successive waves over the next few years. Last year, when I learned of a vacant infectious diseases fellowship position at University of Rochester Medical Center, my regional hospital's quaternary referral center, I applied and was offered the position. Officially entering the field is the fulfillment of an aspiration born more than 2 decades ago when I began reading about disease hunters who traveled the world to study and control emerging pathogens. One of the many perks of joining the fellowship here was having the opportunity to work with doctors Ann Falsey and Ed Walsh, two of the world's top RSV researchers who have spent my entire lifetime and most of theirs studying the virus and helping to make the RSV vaccines a reality. In addition to wanting to express my gratitude for the work you do, I wanted to facilitate an introduction and gauge your interest in having Anne and Ed as guests on TWiV to discuss their work and what they see for the future RSV vaccines and therapeutics. They are as kind and witty as they are brilliant, and I assure you they would be dynamic, entertaining, and insightful guests. Thanks again for enriching my professional and personal life through your expert knowledge, cheerful chatter, and devotion to the pursuit of truth. All the best, Patrick.
Sure, we can look into having them on. That would be great. Sounds good. Angela, you're next.
Do you want me to do the one line by David and then Allison? Because David writes that he thought this would be of interest to us, that there was a measles death, unfortunately, of a child under 5 in Toronto here in Canada,
Right.
which is very tragic.
Also, the one about vaccinating cattle.
Sorry, the David above that. Sorry, I missed that one. There's 2 Davids. Okay, so Dave writes, just listened to a discussion with Richard Webby regarding influenza in cows, TWiV 1013. Unless I missed something, I heard nothing said about vaccinating cattle herds. Possible, available, pros, cons. Thanks and keep up the good work. Dave from Mission Viejo, California. So you're talking about cows influenza? Is he talking about vaccinating cows against influenza?
Yes.
That's not common practice, no.
But how much did we know about cows getting influenza before this?
Right.
Exactly.
I think we talked about this on a subsequent episode. Alan weighed in on it, saying, the development would be substantial. And just like we said for pigs, it's not clear that the farmers would want to do it, right?
Mm-hmm.
I mean, if infection of H5N1 reduces milk yields, then that would be a problem.
Then it might be worth it. Exactly. If it was a significant decrease, because if not, adding any sort of vaccination protocol to any animal in farming is extremely expensive.
Yeah.
Considering the volume of animals.
Could potentially also be an issue with sort of import-export
also
regulations.
Mm-hmm.
Did you want to take the next one, Angela?
Allison? So I already said the other one. Yeah, Allison. Hi, Vincent and TWiV team. First, I wanted to say thank you for all it is that you do. It is such a joy to listen to the podcast. And even as a virologist, I learn something new about viruses every episode. I'm emailing specifically about episode 111. I appreciate how enthusiastic you all are in sharing about the Maya Dam project. It is truly an amazing story with some really exciting experimental design behind the data. In large part designed by a very talented postdoc in the lab, Dr. Wenji Xiao. That said, a few things were incorrect or slightly misinterpreted. We are a lab that specializes in large-scale CRISPR screens, and as such, we generate our own lentivirus and cell libraries. This takes large amounts of work and time and incubator space, and is sort of the bread and butter of Jan's lab. In addition to the exciting science we pull out using these generated libraries. We do purchase plasmid libraries, but from there, we generate our lentivirus and build libraries to have very specific gene coverage. This takes a lot of work, but allows us to feel confident in our screening results. I know you all have so much science to comb through in order to share with the public, but felt this was worth mentioning, especially when others plan or design CRISPR screens for their own projects. Thanks so much for taking the time to read this email, and thank you for all that it is that you do. I truly look forward to each episode. Best, Allie. P.S. Kathy Spindler was one of my committee members for my graduate training, as well as an instructor during my graduate courses. I love hearing her take on new science. It makes me feel I'm back in a committee meeting. Alison Dubzik, she's a PhD or postdoctoral fellow, I should say, Stanford.
So TWiV 1111, we talked about using CRISPR to disrupt necroptosis, and that improves survival after influenza infection. So I had said, yeah, they probably bought the libraries, but this tells us that they make their own. So thank you very much for that. All right, well, we have one more here. Walt writes, I'm always a fan, but specifically wanted to thank your team for the well-organized and thoughtful episode 1121 and the 5 key points excerpt. You often state the importance of basic, who knows where this'll go research. And while this is true also for science communications, it draws a wide range of people to love open-mindedness and inquiry. There's also a burning need to recognize what's most salient to changing the course of the world today and to defend against those who hate open inquiry. I don't think TWiV would be well served by morphing into a broader discussion of the role of politics in science, but even as you did a superb job of sticking to the facts, not insinuations or conjectures in rebutting Dr. Chan and Mr. Ridley, it's important to understand what helps push their ideas to the public. No better example at hand than this recent Washington Post story, how politicians shut down a group that attempted to counter malevolent COVID disinformation, among other topics, and gives a link for that. Thank you again and Godspeed. That's bad, right? They're getting, not only they're spewing nonsense, but they're preventing people from trying to correct them. What are we heading for in this country?
Oblivion.
Come to Canada. I don't know if we're doing much better, but
I think you have some issues in Canada too, don't you?
Nobody's perfect.
But maybe less, I don't know, maybe better.
It's smaller, that's for sure.
True.
No, it's more scenic also.
True. Well, I don't know. You guys have some beautiful
No.
scenery as well.
You've got all of Jasper and Banff. Those are great places.
Also, Angela can go outside today.
Yeah,
true.
That's right.
True. I was sitting outside reading the papers earlier. This is my balcony. I was sitting on my couch.
Sounds
Cool.
good.
All right, let's wrap up with some picks of the week. Angela, what do you have for us?
Okay, so this is maybe spoiler alert, but we all chose like space things except for Vincent.
It works that way.
This was in Nature, actually. So it's by Alexandra Witz, and it is called Staring at the Sun, Close-Up Images from Space, Rewrite Solar Science. So I just thought the coolest part of this was this. So basically when you click on this link, you'll see this solar snake. So they call it a solar snake. It's basically what do they call it? A plasma, a solar snake of plasma appeared across the sun. And there's actually like this little video where you can see, it looks like literally a snake is like I don't know, slithering across the sun. I thought it was so cool. And then they also have another beautiful image of one of the giant solar eruptions on February 15th. It was in 2022, but it's just like a beautiful image about the sun. And they have more information about like research going on, solar researchers. But basically, I wanted to share it mostly because of the solar snake. I thought it was so cool.
Neat.
Yeah.
Brianne, what do you have for us?
So elsewhere in the solar system, I read this article recently and thought it was really cool that there were some scientists who were looking at images of different parts of Mars and suddenly realized that they actually were seeing frost on some calderas of some volcanoes.
What?
This was particularly exciting because these volcanoes are actually in the equatorial region, which is a hotter area of Mars. And so if they're seeing frost there, then that tells you a lot about potential for water elsewhere. They saw this. It is only 1/100th of a millimeter thick layer of frost,
Oh
and
my
it's
gosh.
only there for a very small number of hours per day, sort of in the first time in the morning. And so they really had to luck out to happen to look at images at the right time in the right place. That wasn't what they were originally looking for. But they originally saw it. So they show, for example, an image here where you can see a little bit of frost in a caldera at 7:20 a.m. solar time. In one location. And this was, again, a big surprise, and also leads to some interesting questions about what might be going on with a water cycle in terms of where water has or has not been found, what the hours are of where the water is present, and
Right.
where it's going in other times.
Right.
What does it mean, Brianne?
It means it's cool that we can start to see other places with water, and
hmm
it's also something that, if ever we were to go to other planets or other places, we'd need to have water, and we'd need to figure out whether there's some there or whether we need to bring it or make it. So knowing that, I think they said that if you could collect all of this frost, because these volcanoes are huge, they said that one of the volcanoes was not only bigger than a volcano on Hawaii, but it could fit all of Hawaii in the volcano. So they said that if you could actually get all of that 1/100th of a millimeter thick frost together, it would be 150,000 metric tons of water ice,
Yeah.
or the equivalent of 60 Olympic swimming pools.
That's a lot
Wow.
of ice.
Yeah.
That's so.
Imagine the resolution of their cameras to see 100th of a millimeter.
amazing,
Right.
yeah.
I know.
Yeah, but the
Ice. Isn't
albedo
that
is incredible.
Yeah.
The
It's
albedo
wild.
is
So
everything,
this
right?
means
Yeah, it's
there could be
crazy.
life there as well, right?
Or
Either
was.
now or in past.
Yeah.
Hmm.
And
Cool.
the other thing they worry about is they know there were oceans on Mars at one point because of the geology and the residues. The hematite represents a mineral that's only deposited in aquatic environments. At any rate, they think the water is still on Mars. Where would it go, right? And it's underground. So there are oceans of water waiting to be tapped.
Cool.
So let's all go.
Well, I don't particularly want
Let's
to.
all
It's much
go.
cooler on
I don't
Mars
wanna
right
go.
now than it is
Let's hitch a
anywhere
ride.
You guys can go.
Cheap ride.
What is it?
Oh dear.
8 days to get there or more, right?
Well, they've got a new propulsion system, so it might only take 3 months.
Nope, not happening to me.
Only 3 months.
Nope.
Hmm.
Cool.
Oh well.
Could I bring a comfy couch and my
I
Kindle?
don't think so. You can't bring your cat.
Indeed. No.
Yeah,
Bring a deck of cards.
I can't bring my dog. There's no way I'm going.
It's a lot of solitaire, I tell you.
Dixon, what do you have for us?
Well, another space first. This one involves the Crab Nebula. The James Webb Space Telescope has just published the most resolved picture, a single picture of the Crab Nebula, which represents the remnants of a very large sun, which meant supernova. And the explosion is evident by looking at the trajectory of all of the debris that has come out of that event. When the star exploded, it did so by imploding, and the resultant was a neutron star, which has the diameter of like 1/5 of the Earth, but the mass, if you took a spoonful, it would go all the way from here to China, so to speak. It's extremely, extremely dense. In fact, there are no spaces between the neutrons. They're all stuck together. And the explosion caused an angular rotation, which was maintained by the pulsar. The pulsar is the result of a neutron star turning at a certain rate and emitting radiation from its poles, because in turning it develops a magnetic field. And that magnetic field generates a current, and the current forces particles out at the poles. And so you can see down below, the next picture is an actual combined photograph of the neutron star in the Crab Nebula. That is exactly what it. Now you can't see the neutron star; it's still too small to resolve because it's such a tiny object. But it creates havoc wherever it's found. And the beauty of pulsars, by the way, is what I wanted to just talk about a minute, is that they rotate at a precise time. This particular one rotates at 1.6 seconds. So the entire pulsar turns around at 1.6 seconds.
They're the
But the
best
ones
clocks
that
of the
are
universe. They're the clocks of the universe. Well, my sister uses pulsars also for timing. They're the best timers clocks.
I didn't even know
So
who I
they
was talking in the presence of. I forgot. I
Not me.
forgot.
I'm not. It's Kiana, not me. I don't do that. All I know is
Well,
that they can measure gravitational waves because it disrupts like the
yeah.
pulsar timing.
So they have pictures now of a pulsar that developed next to its binary star companion, and they have a weird relationship also, as most space objects do. The big point to make here about pulsars is that they are essentially astronomical clocks,
Mhm.
and the one which has the most reliable rotation rotates every 33, not hours, not minutes, not seconds, but milliseconds. It rotates once every 33 milliseconds. Can you imagine how fast that actually is? And they can time the pulse, and it's better than an atomic clock.
Mhm.
So they use it for all kinds of measurements and various other things. It's a remarkable. You see the old Star Treks and even the new Star Treks, they avoid all this stuff.
Oh, they didn't know about it.
They don't even know they didn't know about it, but they don't want to know about it. This
That's
is
cool.
really beautiful nature, though. It's actually the symmetry of these things are just incredible. And there's another picture of the Crab Nebula, by the way, for those who are interested in where our elements come from. It's been colorized according to the elements that were ejected from this gigantic star that went into a neutron star. And the amount of, you talk about how much ice you can collect in a volcano on Mars, and it's 100 million, 150,000 gallons of water, something ridiculous like this. The amount of nickel created by the explosion of this star is enough to make a pauper rich in a second and a half.
Hmm. It's such a beautiful picture, the one that's linked.
It's
It
quite
is.
gorgeous. They're beautiful, beautiful objects, but powerful, and you wouldn't like to stand next to one.
I have to add that
It's
since the last episode I was on, Rich told me
yeah.
about the astronomy photo of the day from NASA, and now I have
Oh,
it on my Google Chrome. And now it's
yeah,
like
yeah. Yes,
ideal.
Kathy told me about that, and it is my homepage. So whenever
Me
I open a
too.
browser window, that's what I get. And I feel like there are some days where I just think of random reasons I need a browser window because the picture is so beautiful.
Me too. Sometimes I just leave it open, and at the
Yep.
lab, I have like the second screen. People will come and they're like, "Oh, what is that?" And I was like, "Let's
Yeah.
Now, if
find
I'm not
out."
mistaken, the Crab Nebula was actually the explosion when the star went to supernova. It occurred during humanity's occupation of Earth, and it was recorded by a bunch of cultures, and so we it was quite an event because they claimed that you could see it during daylight.
Cool.
Cool.
Well, I don't know. You guys love space. I know that, but
We do,
I
we
am
do.
more grounded here. I rarely pick a space thing. I don't know why, but the article today is in Science-Based Medicine by Clay Jones, and the title is "Will Your Tattoo Give You Cancer?" Probably not, but maybe.
Hopefully not.
So,
But maybe.
I have like 5. Hopefully
Are,
not.
you have
Tell
a
me, Vincent.
tattoo?
I have like 5. Do you
Good,
guys
good.
have? You got Bran? You don't have any tattoos?
I do
No,
not.
I used to
I've
play.
definitely thought about it.
I used to play baseball, and one day we got tattooed, but that was about it.
anyway,
Vincent should get TWiV.
so why are
Um,
they? So this is based on a study out of Sweden in eClinical Medicine, okay, which showed the risk of tattoos increase your risk of malignant lymphoma by 21%. And I think this is a good example of how you do an observational study and it's just fraught with problems. So why did they do this in the first place? It turns out that tattoo inks have lots of stuff in them, some of which
Sure.
are actually carcinogens, right?
Right.
So this group in Sweden, they took 12,000 subjects. A little over half of them had lymphoma, and 21
Really?
of the subjects with lymphoma had tattoo exposure compared with 18% with no tattoo exposure. Okay. That's where the 21% relative risk increase comes from. And they point out that
That's not
no,
much.
it's not much at all.
Versus 18. Haha.
Yeah,
Okay.
so malignant lymphoma is not very common. The risk is 0.72% in men and 0.35% in women worldwide, compared to 4% with colon cancer, for example. And there are many other risk factors for lymphoma, including viruses and autoimmune disorders and certain other things. But he says that a study like this is like it's an observational study, which probably has all kinds of other um, problems that you didn't pick up, right?
Right.
He makes the point here, which is really very good. He says, where is that? I have to read this for you. Where is it? Where is it? There's this great statement. Hang on. Okay. The confidence interval of the results 0.99 to 1.48. And the author says that a confidence interval that includes 1 does not inspire any confidence in a study
Correct.
like that.
That is correct.
They said there's some other things that we should note. Risk of lymphoma was higher when tattoo exposure occurred less than 2 years before a cancer diagnosis, which strikes me as odd, but also when a first tattoo was at least 11 years old, which makes more sense. Cumulative and higher exposure to mutagens should increase risk, but there was not any apparent increase in risk from having a larger total tattooed body surface area. Finally, some lymphomas appeared, to be more common than others in subjects with tattoos compared to those without. I can't help but worry that this is all, this is all just an example of the Texas sharpshooter fallacy. The most famous example of which, to me, being the uproar over power lines causing leukemia in Swedish children. To be fair, the authors of the study are aware of the many limitations. Many of these assumptions are a stretch. Ultimately, I think we can all agree that it's impossible to put too much stock on an observational and retrospective study like this one, and we need to have
Correct.
better studies, essentially.
Yes.
So this got a lot of
Yeah.
press, but if you read this article, you will understand that it's probably not likely that a tattoo is going to cause cancer.
If someone is super excited about tattoo biology, I'm reminded of a paper that Cindy told us about
Yeah.
on immune about how tattoo ink persists because of macrophage turnover at the site, and the macrophage being unable to digest the ink and turnover. It's a really, really cool paper.
Oh, that's cool.
They pass it on to other
I'm gonna have to
macrophages
read
when they
this.
die. Yeah, it's very cool.
Wait, what?
Yeah.
So the macrophages take up the dye, and then when they die, they turn over the dye to a new cell.
Yeah. Yeah.
That's
They
so
turn into
cool.
melanocytes.
But they do eventually fade. I guess that's more like UV exposure that they fade then, no? Or are they fading because there is a slow digestion? It just takes a long time. I wonder. I have to read this paper. That's cool.
It's in the chat.
We
Thank
have
you.
a listener pick from Jessica. Thank you for years of fun, interesting, and essential education. I thought this little listener pick about an old epidemic might interest you, even though it wasn't viral but bacterial in origin, because of it being one of the first times PPE were used to try and control the spread of an infection. Pictures are interesting too. It's from Jessica, who's an RN and an MA. And this is an article on Wikipedia. The Manchurian plague, a pneumonic plague that occurred in 1910 to 11 in Manchuria, killed 60,000 people, stimulating a multinational medical response and the wearing of the first PPE. Wow. It's thought to have originated with a tarbagan marmot infected with bacterial pneumonia. pneumonia. They were hunted for their fur in Manchuria. It's an airborne disease, incredibly deadly, 100% mortality rate. Very cool.
Wow.
Thank you for that,
Interesting.
Jessica. And that will do it for TWiV 1125. You can find the show notes at microbe.tv/TWiV. You can send your questions and comments to TWiV at microbe.tv. And if you enjoy our work, we would love to have your financial support to keep this going. Go to microbe.tv/contribute. Dixon DePalmier, trichinella.org, thelivingriver.org. Thank you, Dixon.
Thank you, Vincent. Welcome back, and don't try to miss another one, okay?
I won't. I'm here. Brianne Barker's at,
Otherwise, I won't learn a goddamn thing.
oh, you can learn. You'll learn. It's fine. Brianne Barker's at Drew University, BioProf Barker on Blue Sky. Thanks, Brianne.
Thanks. It was great to be here. I learned a lot.
Angela Mingarelli is at McGill University, Immune Vet on X. Thank you, Angela.
Thank you. I had a great time.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.